Yeah.
So basically, because inside, this is for like the dialysis, is that great?
Yeah, this is for the artificial cell experiment.
So because we know that we're changing the amount of solution inside the bag, we know that the mass change will be directly proportional to it because as we increase like the amount of sucrose molarity inside the bag, then that means that the sucrose like, in the bag will want to go out. And we know that osmosis will perform the opposite way, so we know that it will become more like hypotonic, so it want to go more as we increase the sucrose molarity in the bag. So that's what I think.
Good. Yeah, so that was well said. So if we're looking here, the initial experimental data that I gave you was increasing concentrations of sucrose in artificial cells placed in water, right? So in every instance, in the bag that has water in it, surrounded by water. In the bag that has 0.2 in it, surrounded by water, okay? In every one of those except water, the solution surrounding the cell is what? In the black line here. Hypotonic, right? Water surrounding water is isotonic. Thus, you don't see a change in mass. There's no increase or decrease in percent mass. However, when we have 0.2 in the bag and you place it in water, that hypotonic solution, that is just water, is going to encourage water molecules to rush from the outside to the inside, right? Now you're right, the solute of this aqueous solution is going to want to come out, right? So sucrose is going to want to come out, and it will, but you'll still see an increase in mass because of the water rushing in. Now I might, in an experimental scenario, say, well, you know, actually the pores are too small to allow that particular solute to go through, right? Because what is sucrose? Does anybody remember? As far as what type of carbohydrate? It happens to be a disaccharide, so it's two monomers chained together. So you can actually get dialysis tubing that will allow a couple of molecules to go through. Sometimes it's only one, sometimes only glucose, a monosaccharide can go through, right? So I get to define that. But in this case, even if you're getting diffusion out, you're still going to see an increase in mass, a percent mass increase in the time parameters that I'm giving you, right? Water is going to rush in. And it'll rush in increasing amounts when you increase the solute of that solution inside the bag, right? That's why we're seeing this linear trend of percent mass increase with bags containing 1 molar sucrose placed in water increasing the most, right? Now, what's up with that red line? Is it the same experiment? So it's structured the same. We're still looking at artificial cells. There's still some sort of solution inside the bag. There's still some sort of solution surrounding the bag. But instead of water this time, we have what? 0.4 sucrose, right? So you still see the same trend, but you see the data starting off at a different point for cells that have water in the bag. Why is that? What is happening when you place water inside of dialysis tubing and you place that artificial cell into a container containing 0.4 molar sucrose? Max?
The water is going to want to go to the extracellular space because of the sucrose surrounding it.
Excellent. Yeah. So you can say this in a couple of ways. You could say water will go to where there's more solute, and you'll be right. But water is going from where there's more water to where there's less of it in the 0.4 molar sucrose. And that happens to be true for both 0 for water and 0.2. But what happens at 0.4? It's isotonic because 0.4 is inside and 0.4 is outside. So we're being very intentional about showing that line crossing right there where there's no mass increase or mass decrease. Now for 0.42, 0.45, 0.5, 0.6, 0.8, 1. All of those do what? They're on this side. So there's a percent mass increase, which means that water's rushing in, right? Okay. Now, if I wanted to use the same graph for the third part of investigation 4, could I? Absolutely, I could. But it's an entirely different structural experiment, right? Now we have the thing that's constant is the tissue that's going into the liquids, right? Before, the constant was the solution on the outside. Now the constant's the potato cells that we're studying. And we take those potato cells and we put them in different concentrations of solutions like we had before inside the bag, but now those cells are surrounded with either 0, 0.2, 0.4, 0.6, 0.8, or 1. Now, where's that line going to go? Is it the same trend? No. Where does the line start here? Yeah, it basically does this. So as you go from 0, where that solution surrounding is extremely hypotonic, you'll see that percent mass increase. And all of your data suggested is around 40%. And then the line's going to go down here. And then when the tissue is surrounded by 1 molar sucrose, what is happening to that tissue, the potato tissue? It's losing mass because the solution surrounding it is very what? Hypertonic, which is essentially dehydrating potato cells. It's drying them out, right? The water is going from where there's lots of it to where there's less of it in that very concentrated solution surrounding the cell. At each of those points where you're neither gaining, and if you're lined for the potato cells, it's probably right here around 0.6. Those lines where you're not gaining or losing tells you the isotonic state and consequently the concentration of the inside of that cell. Okay, so it's a very simple but very neat experiment to involve quantitative thinking and reasoning surrounding important issues like diffusion, osmosis, and biology. Okay, so you're gonna get some scenarios like this on your test. Not maybe, yes, you will. Okay, so make sure you come and see me if you have any questions on this.